Good morning, good afternoon. Good afternoon and good evening to everyone. My name is Clarissa Marcelle Naidoo, and today I will be presenting my research on the potential inhibition of the human angiotensin converting enzyme 2 and the targeting transmembrane protein series 2 receptors from South African medicinal plants by metabolomic and molecular docking analysis. Just a quick overview or background on COVID. So on February 11, 2020, the World Health Organization classified coronavirus disease as COVID-19. And the coronavirus outbreak was officially declared a pandemic by the WHO on March 11, 2020, almost 4 years later. And why are we still going on about COVID-19? So in 2024, the global population has encountered several health issues that have escalated due to persistent post-COVID syndrome, also known as PPCS. And therefore, discovering novel compounds to manage the enduring effects of PPCS remain a significant priority, especially in South Africa. So this study focuses on potentially inhibiting SARS-CoV-2 HACE and TRIMPAS receptors from South African medicinal plants. So just a quick background on the SARS-CoV-2 virus. So it is a zoonotic virus. It belongs to the family Coronaviridae. In terms of its structure, it's sporoidal crown-like, and its diameter can range from 80 to 220 nanometers. It's classified as alpha, beta, gamma coronaviruses, as well as the delta coronaviruses. Its proteins include the spike S protein, the nucleocapsid protein, the membrane protein, as well as the envelope protein, which all function in terms of attachment, fusion, binding, and acceleration of the virus entry and assembly within the host. Now, what are the key ideas of my study? So specific enzymes such as the SARS-CoV-2 receptors of the study serve as an entry receptor for SARS-CoV-2 to actually infect people. So viral entry requires not only binding to the ACE receptor, but also priming of the virus's spike S protein by the TRIMPAS by cleavage of the S protein at both the S1 and S2 sites. So this cleavage step is absolutely important for the virus whole cell membrane fusion as well as its cell entry. So therefore, the study aimed to discover the SARS-CoV-2 ACE and TRIMPAS inhibitors from South African plant-based products. And the first idea was to scientifically validate the claims provided by the public about the possibilities of South African medicinal plants as potential treatment agents for SARS-CoV-2. And the key idea two was to discover biomarkers that are related to anti-SARS-CoV-2 activities, which can be isolated from natural plant products and can be used for the development of innovative pharmaceuticals, which include possible agents for SARS-CoV-2. So this is just a general overview of the objectives of the study, and this includes collection of the plant material from different geographical locations, conducting chemical extractions on the selected medicinal plants using a variety of solvents, determining the cytotoxicity of the plants against human normal lung fibroblasts, specifically HEL299 cells, performing the metabolomic untargeted analysis using UPLC-MS, and lastly, to conduct the molecular docking analysis and dynamics, which is currently in progress. So just to give you a quick overview of the actual methods, it involved plant collection that was, once the plants were identified and collected, it was confirmed by taxonomists at a herbarium, and the voucher specimens were deposited and conserved, and thereafter the plant samples were transported to the university. And thereafter, the preparation of the plant extracts took place as follows, where approximately 5 grams of each grounded sample of the plant was extracted with about 50 microliters of methanol, acetate, and centrifuge, and thereafter the resulting extracts were filtered using Watman #1 filter paper, and the filter extracts were subsequently used for experiments. So for the cytotoxicity analysis, the cells that were used were HEL299 cells, and they were purchased from the American type cell culture collection. And these cells were maintained using a variety of chemicals and media, ranging from Eagle's minimum essential medium to FCS, bicarbonate, as well as antibiotics, including penicillin and streptomycin. The cells were then grown in a humidifier incubator at 37 degrees with 5 carbon dioxide. And when it was almost at confluency, it was trypanized. Thereafter, the cells were seeded into 96 well E plates, and then they were subjected to the treatment. Afterwards, we had a Resusurian cell viability conducted, and this was read at various wavelengths ranging from 560 to 600 nanometers. For the metabolomics, we had the establishment of metabolites, and the Ultima 3000 LC combined with the Q exactive MS temp functional centrifugation was actually used. And the sample preparation was a bit different from sample preparation for extraction, and it is outlined above. And thereafter, the plant samples were analyzed on both the positive and negative ionization modes to ensure that we had metabolites on both ionization modes, just because sometimes you might have a plant compound that will pick up on the positive mode, whereas sometimes it might only pick up on the negative mode. Hence, we ran it on both ionization modes. For the molecular docking studies on the biomarkers, we used the model basically to determine the interaction between small molecules and a protein. And this essentially allows the characterization of small modules in the binding site of the target proteins, as well as to clarify any fundamental biochemical processes. And the following procedure was used, but I'm not going to go too much in detail for the sake of time. So in the upcoming slides, I'm going to quickly go over the results of the study pertaining to cytotoxicity, metabolomic analysis, as well as molecular docking. As you can see on figure 1, we had the Excelligence real-time cell analysis system, and this allows for the detection of cellular processes by measuring the electronic impedance of sensor electrodes located at the bottom of the wells in the RTCA plates. So by using the RTCA, we can actually take account for the changes in cell number, viability, morphology, as well as cytoskeleton dynamics. And as you can see on figure 1, the extracts exhibited a dose-dependent manner. So this is the results of the Resusurian assay, and it was done basically to confirm the results from the RTCA experiment. And the Resusurian cell viability assay is basically a fluorometric or colorimetric dye that is used to determine cellular metabolomic activity, as well as mitochondrial viability of live cells in a high throughput format. So we use the same concentration that we had used for the RTCA analysis, and all plants extracts exhibited 30% cytotoxicity at concentrations of more than 30 micrograms per mol, signifying their safety on cells, but there were a few exceptions at concentrations of 250 microliters per mol. So for the metabolomic analysis, this was done to investigate the global, we had the principal component analysis and the scatter plot of the PCA model on figure 3. And this was done basically to investigate the global metabolism variations and the principal component analysis in both figure 3A and B. And as you can see in the PCA plot, there's an overview of all samples in the data, and they exhibit obvious grouping trends between the two groups, Sweet Annie and Artemisia afra, which were the study species for this particular research. We then move on to figure 4, which shows the scores scatter plot of the OPL-SDA model in figure 4A. In figure 4B, we have the distribution of the VIP values greater than 1.5. And figure 4C, we have the loading plot of the PLSDA model. So to eliminate any non-specific effects and confirm the biomarkers, we had the orthogonal partial least squares discriminant analysis done. And this actually compared the metabolomic changes in the two groups, Sweet Annie and Artemisia afra. And in the OPL-SDA score plot figure 4A, there is a clear separation of the two groups, which is a bit faint on the slides, but it is indicated of a clear separation. And this significantly changed metabolites between the two groups were filtered further based on the VIP value is greater than 1.5, and then you can see the difference on figure 4B, which shows the distribution of the numerical values. And the OPL-SDA loading plot is showed in figure 4C, and these show the metabolites with significant compounds, and these were filtered according to VIP values greater than 1.5, FC values greater than 2.0, and P values less than 0.05. And these basically show the significant compounds. So figure 5 shows the hierarchical cluster of the metabolome data from the groups Sweet Annie and Artemis Africa. And as you can see, the UPL CMS B chromatograms of the leaves of Sweet Annie and Artemis Africa show the presence of approximately 121 compounds for the negative ionization mode and 81 peaks for the positive ionization mode. And the identified compounds belong to different classes of secondary metabolites, including flavonoids, phenolic acids, glycosides. And from this hierarchy cluster, the highest metabolites in the negative ionization mode was doliclotilene, lyso-PC, and quercetin. And in the positive ionization mode, we actually had oxydiazepine and hydronicotine. So the two best binding energy of the screen phytochemical biomarkers binding at the active sites of the SARS-CoV-2 were expressed in kcal per mole. And table 1 is showing us the docking stimulations of various affinities for the metabolites. And basically, the receptor binding domain of the SARS-CoV-2 receptor plays a very important target for the development of antiviral therapies. So our molecular docking study successfully predicts that the potential compounds, and it also explains the molecular interactions between the SARS-CoV-2 receptors and the two potential ligands, namely doliclotilene, quercetin for the ACE, as well as hydronicotine and quercetin for the tryptase receptors. So the docking results relieve that there is robust interactions with the RBD, exhibiting binding affinities, which have been highlighted in red. And these interactions were facilitated by various non-covalent forces, including hydrogen bonds, pi stacking, as well as van der Waals interactions. So in the year 2020, when the world was faced with COVID, the World Health Organization welcomed inventions from around the world, repurposing drugs, using traditional medicine, and developing new therapies. And as we are still entering 2024 and coming up with new discoveries, we are trying to find out new methods that can actually be used to combat COVID-19 or any forms of COVID viruses in the future. So when we conducted the study, before taking it further, it was necessary to evaluate the cytotoxicity of the plant extracts to determine their cytotoxicity, so we can basically suggest that these compounds that are found via the metabolomic analysis can actually inhibit the SARS-CoV-2 receptors. However, further analysis is necessary to actually substantiate these results. And in progress currently, we have molecular dynamics and in vitro studies taking place. And this is our outline for our molecular docking, molecular dynamic stimulation, and our SARS-CoV-2 enzyme in vitro inhibition assay. And thank you very much.